Hello everyone, my name is Nitish Khurana and I'm a postdoctoral fellow at the Utah Center for Nanomedicine at the University of Utah. The topic of this lecture is Enhanced Permeability and Retention Effect or EPR effect in drug delivery. Now before we dive into what EPR effect is, let's first go through some basic ideas about what drug delivery is, what nanomedicine is. So what is drug delivery? Drug delivery refers to the formulations and the route of administration and the manufacturing techniques and the storage systems and other technologies that are involved in achieving delivery of the pharmaceutical compound to its target site, which is to treat a disease or to achieve a desired therapeutic effect. Now, there can be different routes of drug delivery, how a drug can be delivered to the humans. For example, we are very familiar with the most common route, which is oral, where you take drugs and swallow them like a capsule or a pill. It can be buccal, which means that it can be swallowed or it can be absorbed from the mucous lining of your cheeks. Pulmonary, such as sprays or pumps for your mouth, which directly goes to your lungs. Transdermal, such as the ointments you use on your skin or lotions that you use on your skin. Ocular delivery, such as the eye drops that you use. The route can be nasal, like the nasal sprays that we use. It could be sublingual, which means that the drug can be absorbed if the drug is kept under the tongue, or it could be vaginal or anal, where different ointments could be used to deliver the drug via the vagina or the anal cavity. Now, drug delivery refers to delivery of the drug in any form. It could be a small molecule drug, it could be a drug that is encapsulated in a nanoparticle. Now, what's the difference? A small molecule drug, such as aspirin or penicillin, are very small in size. Their overall structure is usually less than 1 nanometer in size. The problem with small molecule drugs is that they present with a lot of systemic side effects, which we'll talk about in a while. Now, these small molecule drugs can be incorporated or encapsulated inside different nanoparticles. For example, here we have an example of a liposome where aspirin is incorporated inside the aqueous layer of the liposome. Now, liposomes can usually be anywhere between 10 nanometer to 1000 nanometer in size, so it's easy for these liposomes to incorporate multiple molecules of these small molecule drugs inside them, and that helps in a lot of ways that we'll discuss as we move forward in this lecture. So, what is controlled drug delivery? Controlled drug delivery is the delivery of a drug where we can control the rate or the location of the drug that it needs to go to inside of the body over a specified period of time. We can have this control in terms of the temporal delivery, which means that we can have control over the rate at which the drug is released from the nanoparticle, or we can have spatial delivery control, which means that we can have control over the location of where the drug is released. For example, do we want it to be released only in the liver, only in the heart, in any specified organ, or any specified location? Or we could also do a combination of both, where we could direct the nanoparticle to go to a specific location in the body, and we can have the drug released from the nanoparticle over a specific period of time. Now, what are the advantages of controlled drug delivery? Controlled drug delivery increases patient compliance because patients go through less side effects. We need less frequent administration because as we saw in this last slide, one liposome can hold multiple molecules of aspirin, so therefore we can give a larger dose with one single nanoparticle compared to a small molecule drug by itself. We can minimize the local and systemic side effects. Now think about this drug delivery of a small molecule. Now when you take a small molecule, there is a chance that the small molecule might reach all different organs of your body because there is no direction to where the small molecule must go. It just is circulating in your bloodstream, and it tends to reach to every tissue site, and that is what causes a lot of systemic side effects. For example, when we take steroids, we see a lot of side effects like loss of hair, pain in the body in some places. That is because small molecules tend to create a lot of systemic side effects. But we can minimize these local and systemic side effects by using nanoparticles, because we can direct nanoparticles in a controlled fashion where we can have these nanoparticles go to a particular location, and in that way the small molecule drug will only be concentrated, or it will be accumulated in only one position of the body. Other advantages are it improves efficiency in treatment, and it is many times economical compared to small drug delivery. Now let us quickly look at what nanotechnology is. What is a nanoparticle, first of all? To describe nanotechnology, we need to know what a nanoparticle is. So nanoparticle is a subclassification of ultrafine particles with lengths in 2 or 3 dimensions, which are present in the range of 1 nanometer and 100 nanometer, which may or may not exhibit a size-related intensive property. Now here you see a scale of um, different objects in nanometers. So the range between 10 to 100 nanometers is where a nanoparticle lies. Below that, for example, you can see glucose at 1 nanometer size, water at less than 1 nanometer in size. You can see bacteria at almost 1 micron, then cancer cells, and then a tennis ball at this length. So any particle or different nanoparticles that lie between the range of 10 and 100 nanometers are called nanoparticles. Now, what is nanomedicine? The use of nanotechnology or use of nanoparticles in medicine, such as for imaging agents and diagnostics. For example, we use liposomes or different nanoparticles for imaging the tumors in the body. We use real-time assessment to accelerate clinical translation. For example, liposomes are often delivered to humans to see where the tumor is, to map the tumor in the body. We can use nanotechnology in medicine by using multifunctional targeted devices. We can use different nanoparticles, we can put different targeting moieties on those nanoparticles and direct them towards a particular location in the body. We can also do chip-based nanolabs. So these days a lot of chip-based research is going on where you can create organs on a chip so that you don't have to do a lot of research in live animals, and you can do that research that how these drugs and these drugs inside the nanoparticles are going to behave in an organ. You can do that in a on the organ on the chip now. So these are just few of the applications or examples where you can see the use of nanobiotechnology in medicine. Here are some more applications of nanotechnology. You can use nanotechnology in medicine. Like we said, we can use them for clinical investigations. We can use them for surgery. So nanoparticle can be given to patients to map out the tumor, because we can direct the nanoparticles towards the tumor, and we'll definitely discuss more about this as we move along this lecture. But we can direct nanoparticles towards the tumor, so when they accumulate inside the tumor, it helps the surgeon to actually pull the tumor out of the body. We can use them for different occupational and health safety systems. For example, for food and water quality, for air quality improvement, for sensors. So these days a lot of sensors take advantage of nanoparticles in them by using them as diagnostics. We can use them for informatics, for fabrication, for biological constructs. We can use them for homeland security to improve the treatment of infectious diseases. And you have one of the biggest examples in front of you, the vaccine which we are consuming for COVID-19 is a nanoparticle. It's a tremendous breakthrough in the field of nanotechnology, that now vaccines are being delivered via a nanoparticle. We can always improve the environment by the use of nanotechnology, such as we can create better pesticides, so that plants do not the way that we described for humans, how humans can take advantage from nanotechnology because small molecules can reach everywhere in the body. Similarly, plants can also take advantage of nanotechnology when we give them pesticides, in aquaculture, in agriculture, by reducing the pollutants and poisons in the environment. So there are n number of applications that you can think about when it comes to nanotechnology. And I would want you guys to, go back home today and explore some of those applications that you can think of. So some types of nanoparticles. So we've been talking about nanoparticles, the size of what the nanoparticles are. But what kinds of nanoparticles are there that are present specifically? These are for drug delivery purposes. So what kind of nanoparticles are used often, but are not just limited to these 6 examples? It could be a nanosphere, it could be a nanocapsule, where you can have a polymeric shell and you can have drug molecules encapsulated inside these polymeric spheres or polymeric shells. You can have polymeric micelle. Micelle is a single layer lipid structure where you can have a hydrophobic core, where your drug can be incorporated inside the core. One of the biggest examples of micelles is soap. The detergent you use is essentially a micelle. Then the first ever FDA approved nanoparticle, or the first ever nanoparticle to be used in a patient, was a liposome, which was used to treat a cancer. Liposomes are these bilayer structure. If micelle is a single layer lipid structure, then liposomes are bilayer lipid structure, where you have an aqueous core where you can incorporate hydrophilic drugs, or you have this bilayer of polar groups where you can include hydrophobic drugs. You can do all kinds of different multifunctionalities on these liposomes, where you can incorporate antibodies, you can incorporate different imaging agents, you can incorporate different targeting moieties. You also have examples like fourth generation dendrimers. You have a core of dendrimers and you have the surface of the dendrimers. You can incorporate drugs inside the core, in the internal cavity of this dendrimer, or you can incorporate drugs on the surface, which we call them as surface modifications. Then there's this example of a quantum dot, where you use the zinc sulfide shell and ligands over these shells, and this quantum dot is used for imaging, where if you deliver these quantum dots, you can image them at different wavelengths. So now let's look at some of the nanocarriers in clinical use. Doxil, as I said, was the first ever nanoparticle that was approved by FDA for clinical use. It is used to treat, it was first initially approved to treat Kaposi sarcoma, but now it's also used to treat ovarian cancer. So these are not the only 8 examples that you can find. There are multiple examples now. Most of them you would find for different kinds of tumors, but then there are also some nanoparticles that have been approved for, like, vaginal microbicide or hepatitis A, hepatitis C, and right now we have the biggest example in front of us. It's been used against a virus, a COVID-19 virus. Both the Moderna and Pfizer vaccine are based on the application of liposomes to deliver viruses. So let's now go into the EPR effect. Now we've learned about what drug delivery is, what nanotechnology is, what nanoparticles are. So let's see how we can use, and what is the mechanism of these nanoparticles, that they can reach the tumors. How can we direct them towards the tumors, or towards different organs in the body, to treat different tumors that are present in the different organs. So the discovery of EPR effect. Hiroshi Maeda was a Japanese scientist who discovered the EPR effect in 1984. and he suggested this effect called the enhanced permeability and retention effect. His first paper that he uh, like was printed for him was in 1986, and the last paper was in March 2021, because sadly he just passed away a couple of months ago. But I had the opportunity to meet this person because I myself worked on something similar on EPR effect, and you could have not met a better scientist than him. And he just revolutionized the whole field of drug delivery and how we can treat tumors in patients that have cancer. So, what is EPR effect? The enhanced permeability and retention effect, or the EPR effect, is the property by which molecules of certain sizes, typically nanoparticles, tend to accumulate in the tumor tissue much more than they do in normal tissue. Now, let's see what happens here. So, here is an example of a normal tissue, cells present in the normal tissue, and here is an example of cells present in tumor tissue. Now there is a blood vessel that is passing through both of these tissues, right? And we know that the blood vessel is lined by endothelial cells. These are endothelial cells, and you see how these endothelial cells in a normal tissue are tightly packed to each other. And this junction that is present between two tightly packed endothelial cells is called interendothelial tight junction or fenestration. Now, these interendothelial tight junctions between endothelial cells in a normal tissue usually do not exceed the size of 10 nanometers. But what happens in tumor tissue is that these fenestrations or these tight junctions tend to widen up or tend to increase in size, and the endothelial cells start moving apart from each other, increasing these tight junctions between them. Now, why is this happening? Now, for the tumor tissue to grow in size, because tumor has this natural tendency that it wants to grow, for this tumor tissue to grow, it needs a constant supply of water, oxygen, nutrients. So for this constant supply of oxygen, nutrients, and water, it has to pass through these tight junctions. And when there's a constant supply of water and these oxygen and different nutrients, these tight junction gaps tend to start moving away from each other, forming these fenestrations or gaps between them. Now, usually nanoparticles, we work, as I said, in the range of, well, we work in the range of 10 to 200 nanometers, sometimes 500 nanometers. But typically think about this. So these fenestrations, as you can see here, can go for a normal vessel up to 10 nanometers, but up to tumor vessels, it can go up to 137 nanometers, and sometimes even 500 nanometers in some cases. You might be confused of how this morphology is working right now, but I will explain that to you just in a while. So a nanoparticle, say for example of 100 nanometer, is passing through the bloodstream. It cannot cross this tight junction, or in other words, we call this extravasation. Whereas for the tumor vessel, this nanoparticle can automatically extravasate through these fenestrations and accumulate in the tumor tissue. Now you would ask that why would this nanoparticle automatically extravasate or pass this barrier? Because the pressure, the interstitial pressure that is present in the tumor tissue versus the blood vessel is very different. The pressure that is present in the tumor tissue is almost as if it is kind of pulling the particles towards it. So that's why this extravasation happens when there is a presence of these wider junctions. So basically, EPR effect promotes passive drug delivery, where preferentially the nanoparticles will accumulate in the tumor tissue compared to the normal tissue. And this strategy has been used to accumulate drugs in the tumor sites and treat tumors that way. Now, here is a cross section of a blood vessel, which is similar in this image. You can see that in this endothelial cell, it overlaps like this. So this is the This is the tight junction that you are seeing in these two images. So the tight junction between these two endothelial cells doesn't exceed 10 nanometer, whereas in a tumor vessel it can go up to 150 nanometer and even 500 nanometer in some cases. So EPR effect is a combination of permeability and retention, right? Enhanced permeability and retention. The effect cannot survive without one or the other. Permeability means that the drugs have to extravasate through those uh, the fenestrations, and they also have to retain. It means that for a prolonged period of time, those nanoparticles have to retain in the tumor site. For example, in this case that you see, this patient had a tumor in his leg, some mass in the leg, and some in this region, and the nanoparticles that were injected in the patient retained there for 7 days. So it has to be a combination of both permeability and retention. Now let's quickly look at a comparison of blood vessels between normal and tumor tissue. So the normal vessels or the blood vessels in normal tissue are very well organized. They are regularly distributed. They're non-dilated. They're non-permeable. They have a complete basement membrane. Whereas the blood vessels in tumor tissue are disorganized. They're undefined. They're unevenly distributed, dilated, and they have a fractured basement membrane. You can see the difference between normal and tumor vessel here between these two. These are smooth and well organized and regularly distributed, whereas you can see roughness on the vessel here. And in this case, this outer lining is the basement membrane. This is the endothelial cell, this is an actual cross section like we saw in the last slide in the form of a cartoon. This is the actual cross section. So these are two endothelial cells overlapping each other. This is the basement membrane. You can see that in the tumor vessel, this basement membrane is fractured, and this is from a tumor tissue. So we have seen evidence of EPR effect in animals and models. Um, and it's quite well observed. When we grow tumors inside animals, we see that EPR effect is quite prevalent in these animals. For example, in this case, the animal was delivered an imaging agent which was incorporated with a nanoparticle, which is the PSA. Whereas when the animal was delivered just the free imaging agent. So because the free imaging agent is a small molecule, it went all over the body, as you can see, right? All the animal is lighting up because when we put the animal inside the instrument, so basically this instrument, this dye has the ability to fluoresce. So when we put this animal in the instrument, it can read fluorescence. So you can see that the small molecule goes all over the body, and we can read fluorescence all over the animal's body. But when the same imaging agent, which is rhodamine, was attached to a nanoparticle, we see that it preferentially accumulated in the tumors. There were two tumors that were grown in the flanks of this animal, and we see that when the dye was incorporated with the nanoparticle, it did not go to the whole body, but it preferentially accumulated in the tumor. And as you can see in the quantification here, white bar represents the tumor. There was always more uh, dye with the nanoparticle present in the tumor versus in the kidney or the liver. And why are we comparing with kidney and the liver? Because kidney and the liver are the two most important factors that are responsible for clearance or elimination of drugs from our body or even nanoparticles. Then there is evidence of EPR in clinical tumors as well. So here you can see this particular patient was injected with the indocyanine green. Indocyanine green is an FDA approved imaging agent that can be injected into humans. So when the tumor was put into the instrument, it read the fluorescence of indocyanine green, but another patient was injected with indocyanine green, but there was no indocyanine green present in the normal tissue because uh, because it it went into the tumor and not into the normal tissue. It did not go anywhere in the normal tissue. Similarly, in the same patient, you can see that when indocyanine green was injected, you can clearly see a difference of the shade of green color here, that when indocyanine green was injected into this one patient, it clearly accumulated in the tumor tissue, whereas it did not accumulate in the normal tissue. To give you a better example, let's see another patient. So this here is the tumor, whereas this is the normal tissue of a same patient, and this tumor is present in the sinonasal region of this particular patient. So as you can see, when the robot was turned on, and when so ICG was delivered in this particular patient, when the robot was turned on, it read the fluorescence only in the tumor tissue, which you can see in the form of this purple color, whereas in the normal tissue there was no ICG accumulated. And this is evidence of EPR effect, that when we deliver ICG, now ICG you'll say is a small molecule, and I'll talk about this in detail in my next lecture as well, but when ICG is delivered into the blood, it attaches itself to albumin, which is about 5 to 9 nanometers in size, and albumin is the most abundant protein present in human blood. So this ICG albumin complex becomes this nanoparticle by itself of 5 to 9 nanometer in size, and that's why it preferentially extravasates into the tumor via the virtue of EPR effect, whereas it does not accumulate in the normal tissue, because it cannot pass through those junctions. But EPR can be variable. There is a lot of conjecture in the scientific community. Some say that EPR works, some say that EPR doesn't work. For example, in this case, we see accumulation in tumor, but also we see some non-specific accumulation of nanoparticles in other organs. And usually those organs would be liver and kidney, because those are the elimination routes, the natural elimination routes for the human body. So you see that this patient had head and neck tumor, so we see accumulation of the nanoparticle in the head and neck, but we also see accumulation in the liver and in the spleen. This is the spleen and this is the liver, so we see some non-specific accumulation in these organs as well. Similarly for this patient had breast tumor, and this patient was delivered with a nanoparticle. We see accumulation in the tumor, some, but we also see non-specific accumulation in the spleen and in the lungs, and also some in the lymph nodes. So where does that leave us? Is EPR effect real? Is it not real? The matter of the fact is that we don't need to focus on whether EPR is real or not. We know that EPR is real, but the fact is human body is very heterogeneous in nature. Every person, every human body is heterogeneous. Every cancer that presents in a human body is very different. So for example, if I have a particular kind of cancer and some other person has a particular the same type of cancer, our cancers will be very different in terms of how their genetic properties are, how their how the stage of the cancer is, how the grade of the cancer is, what the aggressiveness of the cancer is. So in those terms, human body and tumor itself is very heterogeneous. So we need to basically come up with ways where we can enhance and improve therapies rather than focusing on whether EPR exists or not. We know that EPR exists. We have evidence from the last 35 years. So what we need to do is we need to come up with ways where we can improve therapies. For example, we can enhance extravasation. How can we do that? We can apply heat or hypothermia and open the tight junctions. So hypothermia is the process of increasing the body temperature. We can do that by applying lasers. So if we know that we have a tumor in a particular place in the body, we can apply heat to that particular region, and heat results in opening of these tight junction gaps. And if only 5% of the nanoparticles were earlier accumulating by the virtue of the EPR effect, now by enhancing with the help of hypothermia, maybe 10% can accumulate, maybe 15. So we need to come up with these kind of ways. Well, these are not new techniques, these are old techniques that have been in research for a very long time now. Or we can use targeted nanomedicine. As we saw earlier in that particular animal, the free dye was going all over the body. What if this particular part of the body has a particular gene that represents this organ or this part? If we attach a targeting moiety on the nanoparticle, we can have all the drug accumulated at that particular region, because you must have learned about ligand receptor interactions, right? So if we attach a ligand to the nanoparticle, it can attack this particular receptor and target itself to this particular region, thereby avoiding accumulation all over the body. And these are just some of the ways that have been already employed and have been employed for a really long time in research to improve So with that, I'd like to go over some concluding remarks and future directions. We know that EPR effect exists. It is variable from cancer to type, depends on the stage of the cancer, it depends from patient to patient. What we need better animal models, and that's the future direction. We need to have better animal models where we can justify EPR effect being or we can, have the debate whether EPR is applicable in this animal model or not, because right now we see EPR present in every animal model, but that's not true when it comes to the um, human clinical trials. We need methods to enhance EPR effect. We've already used hypothermia, vascular, or tumor targeting. What we need is personalized medicine. If my tumor is representative of my human body, then we need to design drugs based on my body, of what the genetic expression of my body is, and we need to come up with ways to use those genetic markers as targeting moieties and come up with drugs, which is called personalized medicine. So that's what we need to do moving into the future. And we need to come up with combination strategies where we can combine personalized medicine with other aspects like tumor targeting or hypothermia, and these combinative or this combinatorial effect can lead to improved efficacy. And with that, I'd like to thank everyone. Thank you.